Chapter 6 is all about the cell structure. So we introduced in Chapter 1, what's the bare minimum requirements for a cell? Well, you have to have a plasma membrane, you have to have DNA, and you have to have ribosomes, and you also have to have that fluid-filled space, which is a mixture of water, proteins, nucleic acids, carbohydrates that makes up the cytoplasm. Simplest level, there are two basic kinds of cells. You are a eukaryote. So your cell has all of these parts, but in addition to them, eukaryotic cells have membrane-enclosed organelles, which is the major concept we're going to talk about in this chapter. The most obvious of which is the nucleus. Prokaryotes, on the other hand, are things like bacteria and archaea that lack membrane-enclosed organelles. That means they don't have a nucleus, they don't have a mitochondria, but all cells do have these four structures in common. So we'll just talk a little bit about prokaryotes. They are a lot more simple than our own cells. So they have these four structures that all cells have: plasma membrane, DNA, ribosomes, and cytoplasm. Since they lack membrane-enclosed organelles, their DNA just floats around in the cytoplasms. In some species, it just floats in the center, in others, it's pushed against the plasma membrane. And this clump of DNA is called the nucleoid, or the nucleoid body. Some structures that prokaryotes have that are distinct from animal cells, in addition to the plasma membrane, they have a structure outside of the plasma membrane. Okay? And this structure outside of the plasma membrane is called the cell wall. Animal cells don't have a cell wall. The cell wall is not the same thing as the plasma membrane. In bacteria, the cell wall is made out of proteoglycan. It's a mixture of proteins and carbohydrates. Some species of bacteria have another layer outside of that called the capsule, which is a sticky coating of carbohydrates that helps them stick to each other and stick to surfaces. Some species of bacteria have flagella that allow them to move, although these are distinct from the flagella found in eukaryotes. And others have small fibers that allow for exchange of genetic material called pili. Or the fimbriae are short little hair-like structures that help them attach to other substances. So that's about all we're going to say about bacteria for now. You do need to know the major difference between eukaryotes versus prokaryotes, and what structures are unique to bacteria versus animal cells. So if we look at a typical animal cell, the first thing that I want you guys to recognize is this is an artist's rendering of a eukaryotic cell. No cell looks exactly like this, and no cell at all has a blue endoplasmic reticulum and a green Golgi body and orange mitochondria. They've just made them different colors so that they're visually distinct from one another. So what I have done on this slide is highlight a couple structures that are unique to animal cells. Animals are eukaryotes that are not found in plant cells. Plants are also eukaryotes. Okay, so the ones that I want to point out here, the lysosome we'll talk about is not present in most plant cells. In most plant cells, it's replaced by the central vacuole. Centrosome, which is made of two centrioles, is found in animal cells, but is not found in most plant cells. And we'll talk about the function of each of these structures in a little bit. So the other thing that I wanted to point out is which structures are unique to plant cells that are not found in animal cells. The most obvious of which are the chloroplasts. The chloroplasts are the structures that do photosynthesis. I want to point out one structure that is present in plant cells and animal cells. Write this down. Plants have mitochondria. Plants have mitochondria. They have mitochondria and they have chloroplasts, but plants have chloroplasts and animal cells do not. A couple other structures that are unique to plant cells. Plants have a cell wall outside of their plasma membrane, but the cell wall in plants is distinct from the cell wall in bacteria. In plants, it's made of that carbohydrate cellulose that holds them up and gives them structure. That cell wall is actually perforated with holes that allow for the flow of cytoplasm, sugars, even ribosomes and RNA between plant cells. And those holes are called plasmodesmata. And then one other structure that's unique to plant cells, the central vacuole can be a storage organelle and also just takes up a lot of space in the plant cell. So we're going to talk about each of these structures as we go through this chapter. So I want you to think, what do you think is the most important structure in a cell? So I'm going to put the animal cell back up here and just jot down, what do you think is the most important structure? Go ahead and pause the video and do that now. If you picked, say, the nucleus for the most important structure, well, prokaryotes don't even have a nucleus, okay? And in your own body, your red blood cells actually lose their nucleus during differentiation, so it can't be the nucleus. Ribosomes, again, pretty important. All cells have them. You can't get along very far without them, but I would argue that the most important structure in a cell is the plasma membrane. The plasma membrane is what distinguishes the extracellular environment from the interior of the cell. It's what makes the cell the cell. Plasma membrane is made up of those phospholipids that we introduced in the last chapter. Phospholipids, remember, are amphipathic. Amphipathic means that they have a hydrophilic head group and a hydrophobic tail group. Amphipathic molecules will self-assemble into a phospholipid bilayer. Amphipathic molecules will self-assemble, so the polar head groups will react with water on the outside of the cell and also in the cytoplasm, whereas the hydrophobic tails will try to get away from the water and clump together. And this phospholipid bilayer makes up the cell membrane. Also describe the cell membrane as semi-permeable, meaning it allows some things through and prevents other things from moving across. Molecules that are very small and non-polar can actually go directly through the phospholipid bilayer. So oxygen and carbon dioxide can move right in between these phospholipids. Small polar molecules like water can also squeeze through, but the larger the molecule is, the harder it is for it to squeeze through. So large polar molecules like sugar have trouble going directly through the membrane. Charged particles, ions, can't get through the membrane, and those need some help getting across. So there are also proteins that make up the cell membrane, so they're embedded in the cell membrane. And there's lots of different functions that these proteins have. Some of them make channels that allow for transport of molecules like ions into and out of the cell. So one more way to describe the cell membrane is we can describe it as a fluid mosaic. Each individual phospholipid and protein is like a tile in a mosaic, but unlike a tile mosaic that's glued together with cement, each individual piece can slide past one another laterally, so they can slide around and move past one another. And so it's more like a glass of water than it is like a brick wall. And that's part of why the plasma membrane is semi-permeable, because the adjacent phospholipids are not permanently glued to each other. Remember, lipids don't form polymers. The only thing that's holding those lipids together is hydrophobic interactions, where the hydrophobic tails want to get away from water and the hydrophilic head groups are pointing out towards the water. We'll talk more about transport across the membrane in the next chapter when we begin Unit 2. So one more concept that I want to introduce with cells is this fact that the plasma membrane regulates everything that comes into and out of the cell. Because of that, the plasma membrane actually limits how large cells can get. So if you imagine you have a cell that's 1 by 1 by 1 units in length, width, and height, and it's a perfect cube, okay? Then each side of the cube has a 1 square surface area, okay? And so since it's a cube, it's got 6 sides, so it has 6 total surface areas. And the volume is 1 times 1 times 1, so it has 1 cubic volume. So a small cell has a very large surface area compared to its volume. But if we make that cell bigger, let's say now it's 5 by 5 by 5 units, the surface area does go up, right? And the volume does go up, but the surface area is going up more than the volume, okay? So now we have 150 surface area units compared to only 125 volume. And so now the surface area to volume ratio is only 1.2 compared to when it was 6 surface areas per volume when it was a small cube. So organisms have very small cells to solve this problem. Small cells will have a high surface area to volume ratio, which allows for sufficient nutrients to get in and sufficient waste to get out. All cells aren't perfect cubes, so there are different strategies for maintaining this high surface area to volume ratio. So I want you to imagine that you have a ball of Play-Doh in front of you. Okay? What could you do to that Play-Doh to give it a large surface area compared to its volume? So one thing you could do is break it into tiny pieces, and that would be the strategy of having very small cells. So go ahead and pause the video and think of what else you could do to accomplish a high surface area to volume ratio. So here are just a few ideas. So one thing you could have done is fold that ball of Play-Doh back and forth. So this is a strategy that's taken by our gut epithelium. These little finger-like projections called microvilli increase the surface area without changing the volume of the cell very much. Okay. Another strategy employed by plants is to have a large central vacuole. And that vacuole is basically metabolically dead. It doesn't need nutrients, it's not making waste. And so the cell can be bigger because it has this big empty bubble in the middle. Our epithelial cells have a large surface area in 2 dimensions, but they're pretty flat. So this is the top view, and then this is the side view of, say, a cheek epithelium. And so even though they have a lot of surface area in 2 dimensions, since they're very flat, they don't have a lot of volume. And then this other cell, which is going off the screen, is supposed to be a neuron. The largest cell known is the neuron that goes from the hip to the toe of a giraffe, and these can be up to 6 feet long. So again, they're very large in 1 dimension, but as we cut across here, it has a small volume at each place. So cells still have to keep a large surface area to volume ratio, even though they come in a variety of different shapes.